So, welcome everyone. This is my presentation, the generation of near-infrared genomic silver clusters via attribute regularized variational autoencoders. And this is the topic that I've been working on with Professor Bogdanov at the UAlbany Data Mining and Management Lab. I recommend definitely looking into some of his work, or maybe even reaching out to him if you're interested in machine learning at UAlbany and want to get some research experience. He's definitely the guy. So my talk is basically going to be about what exactly we're doing, the kinds of problems we're trying to solve, and then a large portion of it is going to be focusing on the methods that we're using to solve those problems. So let's start things off. So here's the overview of basically what the presentation is going to cover, right? We're going to do a quick overview and the goals of the project. Then we're going to cover the big picture model of how the data actually flows through. Then we are going to talk about, and everything else is just part of that. So this is all part of the big model on how we're actually doing that generation. And it's all the data flow, right? So, oh yeah, there's also there's like a presentation within the presentation here. Part 4, an introduction to AR-VAE. This is the newfangled machine learning model that we're using. It's an attribute regularized variational autoencoder. And this is very powerful. This is a very powerful generative model that we've found is actually pretty good at creating these DNA sequences the way that we want them. So it was based actually on these people trying to create, they were trying to generate new music pieces. And we kind of just took that and we're like, hey, we could use this to generate DNA sequences that have properties that we want. So it's a really cool model. If you take one thing away from this presentation, I hope that it's just how cool this model is. So let's move forward here. So overview and goals. First things first, understanding a bit of the terminology, right? Genomic silver clusters. So genomic silver clusters, which I'm also going to be referring to as GSCs, and in some of the papers on them, you'll see them referred to as AG sub N DNA. They're basically nanoparticles of DNA and silver, and they fluoresce when they're exposed to UV light. Now, fluoresce, I know we're not chemistry or physics people. Fluoresce just means that it emits light when you shine light at it, right? So I think this is probably wrong, but I think glow sticks like fluoresce, right? Like fluorescence, they let out light. So basically, the way that you create these, you have a little snippet of DNA, about 10 DNA bases. I assume I don't need to explain what DNA is, right? Like everyone took like high school biology, right? We all good on DNA? Yes. So we have like an open, unzipped strand of DNA, and we put a bunch of silver ions in solution with it. And then there's some chemical reaction that happens that I'm not on the chemistry team. We're collaborating with chemists and material scientists at UC Irvine who actually do all of the chemistry to make this and make these reality. I'm just doing the machine learning that actually generates these, right? So essentially what happens is we have this like 10 base sequence of DNA, and it's in solution with a bunch of these silver ions, just standalone silver atoms. They do some chemical reaction on it. And then what happens is the DNA wraps around these silver ions in a specific way. And when it does that, this new complex may or may not fluoresce based on the structure of it. So basically, when we shine UV light on these nanoparticles, it fluoresces and lets out light of a certain wavelength. So here's where things get really cool, is that not all of the clusters are created equal. So essentially, different DNA sequences produce clusters with different intensities and wavelengths of fluorescence. As in, depending on your DNA sequence that you're using to create these with, you're going to have a different wavelength of light. So some might fluoresce green, some might fluoresce red when you shine UV light on them, based on the sequence of DNA bases that you're using to construct these nanoparticles. So essentially, a previous study that the DMM lab collaborated on before I was in it basically found that different bases and motifs have effects on the intensity and wavelength of the final particle based on those DNA sequences. So one of the things that they found was they did a bunch of this motif recognition, where basically they found that different letters of the DNA sequence correspond to different colors. So if your sequence has a lot of T's, it's probably not going to fluoresce at all. It's going to be dark. If your sequence has a lot of C's and A's and a few G's and T's, then it's probably going to be green, right? So on average, we see these patterns. If a sequence has a ton of C's, it's probably going to be red. And if it has a lot of C's and G's, it's going to be very red. And essentially, we can look at sequences and basically eyeball what we think they're going to be, right? So if I see a lot of T's in this sequence based off of this chart, I would say, okay, this sequence is probably going to be a dark sequence. When we actually turn this DNA sequence into a nanoparticle and shine light at it, it's probably not going to fluoresce. It's going to be dark. If we see a lot of C's in something, we're probably going to assume it's either red or very red, right? And if we see a lot of A's in something, like this final one here, I think this one's supposed to demonstrate A's, we might assume that it's green, or we might be able to assume that it's dark, right? So in general, we're able to basically eyeball what we think the color is going to be. But that's not 100% true 100% of the time, right? So this is just in general. And we want an even deeper understanding of why certain base compositions fluoresce a certain color. So, any questions about this? We're still on like the basic chemistry stuff of what's going on here. We're just creating these little nanoparticles with DNA, and essentially the color that we fluoresce at, the wavelength of the light, is dependent upon the sequence of DNA bases that we pick for our particles, right? I hope that makes sense to everyone. So, moving forward from here, our objective on this project, I see someone typed in workshop text, basic. Yes. So the objective on this project essentially is we want very special sequences that are in the near infrared wavelength region, right? So I don't know how many of you know about the visible light, the light spectrum, right? There's the electromagnetic radiation spectrum. You have your visible light in the middle, then you have your infrared light, and going in the other direction, you get like ultraviolet light and then gamma radiation and stuff like that. Is everyone familiar with the electromagnetic spectrum? I think it's like taught in most high school physics classes, but essentially, we're just going to assume that you don't even have to know about the spectrum itself, right? So we're looking essentially for clusters that when you shine UV light on them, their fluorescence is going to be infrared fluorescence in this region right here. So what we're classifying as near IR is 750 nanometers to 1400 nanometers for the wavelength of the light, right? So the thing is, this area of the spectrum is really useful for applications. So we're talking like, so essentially because these are DNA particles, right? So what we can do is we can actually take these and bind these to other pieces of DNA, potentially malicious DNA or whatever, like different viruses and stuff. And then if we shine UV light through the body, then we can ideally identify. So say you have a cancer strand or some virus inside of you that has a known amount of DNA. We attach a little piece of DNA to the end of it that will bind to a marker on the malicious DNA. Then we shine UV light through you, and then it fluoresces in near IR, which will go through your skin, and thus we are able to basically identify where cancer is, where malicious actors in the body are based off of this. So that's my understanding of how that works. I have no idea how this works biologically. I'm not a biologist, but that's my understanding of one potential application of this. When I first started, I thought we were like designing this as like a cheap glow stick solution, and we were going to be able to make some really cool glow sticks out of it. Unfortunately, it has some serious applications. Well, I should say fortunately, because they were kind enough to give this a nice grant because of those potential applications, right? So there's a lot of good, useful applications for these near IR fluorescence DNA nanocluster particles, right? So basically, here's the issue. They're very hard to find, at least the ones that we want. Most of the fluorescence that we have is in this near IR, is in the visible light spectrum. So it could literally be green that we can see, red that we can see, very red that we can see. We want near infrared, which is stuff that we can't see. Of 1,432 samples that we tested, only around 10 of them were near IR sequences. And as you can, this is the distribution of all of the sequences we tested. So the data set here is 14,000 or 1,432 samples, and we can see just how few of those are in the region that we want. And ideally, we want ones that fluoresce even more near infrared than what we have right now. So that's the core property that we want, right? We want them to be fluorescing in the near infrared wavelength. But there's more than just that. We also want beyond wavelength. So essentially, these are like literal molecules, right? We're designing these molecules. So the other thing that we care about is the fact that they're chemically stable in whatever solution they're used for applications for. So for example, if you're going to use this as like a tag and to identify things in the body, you want it to be able to exist within the human body. And that's an environment, right? There's temperature conditions, there's pH conditions, stuff like that. So we want our clusters to also have chemical properties that make them stable in whatever environment they're going to be used in. Because if they just deteriorate outright the second you put them in the application environment, well, too bad, we're not going to be able to use them then, right? So that's another property that we want to design for, right? So our whole goal is that we want to basically be able to design for these clusters with these desired properties. We want near infrared, we want to be in this area of the electromagnetic spectrum, and we want it to be chemically stable. That's like our big two concerns that we have, right? And we also want it to be bright, right? Like, so even if it's in the near infrared spectrum, it could also be in the near infrared spectrum, but also be very dark, which we don't necessarily want. We want something that's really bright, where you shine UV light at it and it gives off a bunch, it reflects basically a bunch, it fluoresces a bunch of near infrared light back at you, right? That makes it much easier to identify. So, moving on here, let's talk about the problems with actually trying to find these, right? So, genomic silver clusters, essentially the biggest issue here is the fact that they're very small. So it's almost impossible for us to perform physical analysis on them on why certain sequences are going to be fluorescing a certain way. These nanoparticles are very complex structures. Like, they're at the level where they're big enough that it's kind of hard to reason about why exactly it's fluorescing a certain wavelength based on a certain strand of DNA. So, long story short, that's the biggest issue with... So we can't just go in, look at it under a microscope and say, ah, yes, it will fluoresce this color because it has this geometric structure. It doesn't, unfortunately, work like that. There's no way that we're able to do that. They're too small to perform like any real physical analysis on of why they're fluorescing a certain color. The other issue is the fact that it's almost impossible to brute force this. When you think about how many DNA bases we're able to do, even with the chain of just 10 bases, there's over 1 million permutations of 10 DNA bases. So in our case, we're using a chain of 10 DNA bases, but you can construct this out of arbitrary length chains of like 30, say, bases, right? It's to the point where if you have a chain of 30, then you have over 10 to the 18 possible DNA base combinations for your chain. And because we're not just simulating these, we actually have to construct them in real life and test these out on real weld plates, and that's very expensive to do. It's not feasible to brute force all of these different combinations to see which ones work. So if we look back on previous slides, our sample set was only 1,432 different base pair, different sequences, right? So it's not feasible that we would be able to actually physically test in real life over 1 million different sequences, even with some of the technology that we have, because it's, again, very expensive to sequence these. So basically, this leaves us one option left. And that option is to use machine learning, which, again, is a statistical technique where we're learning from the data that we do have underlying properties that will help us figure out what we want. And in our case, we're trying to design for these features that we want. We want to be able to design for high brightness, high near infrared wavelength, and decent chemical stability, right? So in our case, the actual method that we're using is going to be a generative model, and it is called our model is a variational autoencoder. It's a special type called a attribute regularized, I can't pronounce that still, variational autoencoder. I'll refer to it as AR-VAE to save you from my terrible pronunciation of it for the rest of the presentation. And it's, in particular, we've customized it to use something called an LSTM network, which it's, if you were at the presentation last night, you'll remember that those are very good for sequences. And we just so happen to be operating on sequences, DNA sequences of 10 bases. So that's why we are using a long short-term memory network for our encoder and decoder, which will make a lot more sense when I go over this in the next portion where I explain what exactly a AR-VAE is and how it works. So let me jump right into the big picture model overview for how the data actually flows through our big machine learning network that we have going on for us. So let's actually talk here about what's going on. So essentially, we start off with genomic silver cluster synthesis and dataset generation. So long story short, we pick a bunch of random sequences, a bunch of random DNA sequences of 10 bases long, and we create them in real life. And once we create them in real life, then we test them for what properties they have. This is done by our collaborators at UC Irvine. They have a robot that basically creates thousands of these at a time. Not thousands of these, like hundreds of them at a time. And essentially, they actually have a physical device that then shines infrared light on these sequences. And once we do that, we are able to actually get what the wavelength of that particular sequence is, and we get the intensity. And we aren't testing chemical properties yet, but we sure will be soon. After that, so yeah, we record the attributes that we want. We write the values to the data set. And then that gives us a nice data set to use. And once we have the data set, we are able to use our machine learning model to learn from that data set and hopefully learn well enough that we can do data generation as well and generate new sequences with our desired properties. So next, we pass in the data set that we've just gotten and the hyperparameters, and we pass that into the model, and we train the model. And once the machine learning model has been trained, we do a bunch of analysis. And out of that, we get metrics. And with the metrics, we are able to basically judge how well our model actually learned the data set. And that's what we care about. So we essentially, there's a lot of other parameters for our model. These are the hyperparameters. Hyperparameters in machine learning are parameters that impact how well the model does, essentially. It impacts the model itself. It can impact the shape of the model. I'll be going over the hyperparameters in detail in a later section. And essentially, the hyperparameters are just other values that are going to be impacting the training, and it's going to impact the metrics as well, and just the performance of the model in general. So because of that, we pass in a bunch of different hyperparameters, and essentially we save each model and all of the metrics for all of the runs. And we actually do this a bunch of times with a lot of different hyperparameter settings. This is called a grid search because we're looking at a grid of parameters, if you will, being the hyperparameters, and trying out a bunch of different values for those hyperparameters. And because of that, we're able to actually get a whole grid of models with separate metrics. And then we go in as humans and look at what the optimal metrics are and what hyperparameters are causing us to have really good metrics. And then we will take the model that we think is the best model, or we'll take a look at the grid search and say, oh, okay, it looks like if we push the hyperparameters in this direction, that the model performs how we want. Let's try more and do a better grid search in this specific area to see if we can find even better hyperparameters to give us even better metrics. And then once we've done that a lot of times, then we are able to basically deduce, okay, with the model that we have, this is going to be the optimal metrics that we can get. And then we denote that as the optimal model. And once we compute the optimal model, then we are able to actually use our model for its generative properties. And this is where AR-VAE comes in. This is what makes AR-VAE so powerful, is that AR-VAE is a generative model, which means we are able, using AR-VAE, to basically select what attributes we want, and then it will spit out a DNA sequence with those attributes. And that's what is really important here. We are able to, once we get a model that is good, we can basically we can take the decoder out, which is what converts the attributes to a sequence, and then we are able to essentially manipulate the whatever attributes we want, and hopefully it will spit out a sequence that gives us very high in real life properties that we desire, right? So if we say, hey, let's create a sequence that is near IR and give it really high, give it a near IR sequence, and say, okay, we want this to be a near IR sequence, and we want it to be chemically stable here, here, and here, it will then spit out a sequence that is hopefully chemically stable and in the near IR spectrum, right? So because of that, so finally we manipulate the optimum model to produce the sequences. That's what I was just saying. Yeah. So next up, this is basically the small picture. Does anyone have any questions about like this? This is a general overview of the data flow that we have going. All right, no questions. Moving on to the real data flow right here, which is the bigger version of that, which we're going to be going over in much more depth. So moving through this, the first thing that we're going to cover is basically the actual process of synthesizing the data cluster, the silver clusters, and then generating the data set from the synthesis. So a lot of this is going to be a bit chemistry-y. So we're going to go over the entire process here of what's actually going on. So we start off with our list of DNA sequences that we've randomly selected, 10 bases, right? So we send that in, and then we also send in what attributes we actually care about. And then essentially, a lot of humans are doing this, so this isn't really a data flow. This is like actually testing, right? So the first thing we do is we take our DNA sequence and we actually create it in real life. We synthesize the DNA sequence with the silver clusters, and then we get a genomic silver cluster. We then shine UV light on that silver cluster, and out of that UV light, we get this fluorescent light, right? And then a spectrometer / fluorometer reads that fluorescent light and gets a spectrum, right? And then we have this basically spectra right here, right? We have where the it's kind of hard to explain how light spectra works, but essentially, normally we're going to get a Gaussian distribution for the light. If you've ever seen like the light coming in from stars, this is kind of similar, where there's just Gaussians of the light and how much light gets in. So we normally consider the wavelength to be just the center of the Gaussian, right? So we'll see that more in the next on the next slide. So basically, the fluorescent light comes off at different wavelengths, but primarily at one wavelength, right? And then with that, we actually can take the area versus the area under the intensity versus wavelength curve. That's our integrated intensity. If anyone's taken calculus, this should make sense. Integrated intensity. This is the intensity versus wavelength. The integrated intensity is just the area under this curve, right? And then the wavelength at which the curve hits its maximum intensity is its wavelength of the peak, right? That makes sense to everyone? Wavelength of the peak, area where it hits the top, it's the argmax of this curve, and then integrated intensity is the area under the curve, right? So those are two of the properties that we care about controlling, because we care about how bright it is, and we care about essentially what wavelength it is. We want it to be in the near IR spectrum. And then finally, we're also going to be doing chemical stability testing on the cluster, and we would put the chemical stability metrics in there, which we don't have at the moment. And all of this comes together. We put in this original sequence, the chemical stability, the integrated intensity, and the wavelength of the peak, and this becomes a single data point that we care about. And then we do this a bunch of times. We repeat this process for all of the DNA sequences, and that is our data set. So ideally, we have like 2,000. I think in our current data set, we have 2,134 of these. So of these data points that have been physically constructed. We've figured out what the integrated intensity and wavelength of the peak is, and we have a nice data set of that. And then we use this data set that we've constructed physically to train our machine learning model. So let's go take a look at the actual, oh, that's right. I have a really nice thing here in what's actually being stored inside of the data points, right? So the DNA sequence is just the string of 10 DNA bases. That's basically our label, right? That's what the DNA sequence actually is. Then we have the attributes, right? And so first off, the chemical stabilities, but red attributes, we haven't implemented them yet. Everything else we have implemented at the moment. So we're doing integrated intensity and wavelength of the peak right now. So these diagrams basically just show off what these properties actually are. So when you get the fluorescent light back, you get the intensity versus wavelength curve from the fluorometer. And to get the integrated intensity, we get the area under the curve. To find the wavelength of the peak, it is literally just the argmax of the function, and it's just the peak. It's the location at which, it's the wavelength at which this curve reaches maximum intensity, right? Makes sense? And essentially the way that we store this information, since normal distributions, they're basically parameterized by a mean and a standard deviation. So we literally just take the mean that this is parameterized by, right? So yeah, moving on. Any questions about what's in a data point, by the way, before we move on? Good. Okay. Excuse me. Okay. So before we move on, I'm going to be doing an introduction to the AR-VAE model and why this model is so exciting as compared to other models. This is a really powerful generative model for generating data once you have data. Essentially, it was introduced in this paper, attribute-based regularization of VAE latent spaces, and it's again, it's a very, very powerful model. So before we go into what AR-VAE is, first we really have to cover what normal autoencoders do. So essentially, autoencoders are a type of machine learning model that consists of 3 different parts. We have an encoder, a latent space, and a decoder. And the goal of the autoencoder is to basically reconstruct the original input, and in the process, it learns what low-dimensional data corresponds to our data set. So in this case, this is a good model of what an autoencoder looks like. You have your encoder, and inside of this encoder, could be any machine learning model you want. So for example, if you guys remember from, I might skip ahead a slide to just show off what they actually look like. So this could be the machine learning model that we're using as the encoder. This could be the machine learning model that we're using as our decoder, and this is our latent space, the compressed space in the middle. So again, don't worry too much about this other slides. We'll get there eventually. But essentially, this is also called the latent space or the latent code for a single object that's been encoded. And basically the goal is, again, we have to learn what low-dimensional data and what sets of attributes correspond to our data, right? So if we're putting a 0 in, we're basically compressing it to its attributes, and then the decoder is basically uncompressing that. And the goal is to have the output match the input. That's how we tell how well we've done. So basically what ends up happening is, hopefully, the encoder would encode this face into his properties, right? His smile, his skin tone, his gender, beard, glasses, and hair color. And the decoder would take these attributes and reconstruct the face from that, right? So this is where things get really powerful, because we're all of a sudden, we have the ability to encode and learn arbitrary properties from this and then decode. So here's the issue with this, right? This seems really nice, right? Like, we have the ability to read out specific properties of this guy and then recreate that image perfectly, right? And that's what autoencoders are supposed to do in theory, right? And theoretically, we should be able to change any of these properties in the middle here and have it output a different face and generate a completely different face. But here's the issue with this, is that's unfortunately not how it works. So in reality, the dimensions in the land space, they're not actually separate. So smile, skin tone, gender, beard, glasses, hair color, the attributes are all entangled with one another, right? So a 0.99 in that first dimension, it actually might correspond to multiple things of these, and it might all be entangled. So a 0.99 in the first dimension, it might correspond to a medium smile, large beard, brown hair, all at the same time. So all of these properties are actually all tied up with one another. And that's terrible, because if I change what I want to be the beard dimension, I don't want it to be affecting his hair color or his skin color or the person's gender, right? That completely ruins the entire point. We want to be able to manipulate these attributes independently. We don't want these to be entangled. And as we're going to see, first of all, this just isn't a good generative model because it's discrete. But even once we make it a good generative model, we're still going to have this problem. And AR-VAE really focuses on building on and fixing this problem, so we're able to have a single attribute in each of these latent dimensions that we're able to manipulate independently. So let's keep moving forward here. So this is a 2D latent space of an autoencoder on handwritten digits. So in this case, they're compressing it down to 2 different dimensions in the latent space. And so as you can see, we encode, right? We have all of our data. We encode it to a latent code. And in this case, our latent code is 2-dimensional. There's 2 different variables that we have access to in the latent code. And in this case, they're just plotting these, right? They're plotting Z1 and Z2 on a 2D plane, right? So essentially, because of this, we can see that in the latent code, this is basically all of the different data points from before, all of the different data samples, all of these different handwritten digits are being mapped to one single point in 2D space. And we see that essentially what happens is they group up in the latent space as the model learns, right? So it's placing all of the 1s right here. It's placing all of the 7s right here. It's placing all of the 6s right here, all of the 0s right over here in this latent space. Thank you, Connor. And what's going to happen is the fact that the bigger issue is when we want to design when we want to basically create a new number out of this, we want to actually be able to create the number. But what ends up happening is places like right here exist, where if we set our 2 attributes that we had access to, which, again, these attributes don't actually mean anything, the attributes Z1 and Z2. In this case, these attributes have meaning. But what ends up happening with normal autoencoders is that the attributes have no meaning whatsoever. They're not significant to humans. They're only significant to the model. They might correspond to lots of different properties, or just no properties at all, or some really high-level property that only the model understands that no human would ever be able to understand. So this is all entangled. It's all messed up. All we can see is that apparently they've all grouped up, right? So if we assume that if we put something in here, we'd be able to get a 1 out. But if we set the 2 attributes in the latent space to something like this question mark point right here, it would have no idea what to give us. And we would get some nonsensical thing out of the autoencoder. This is not a good generative model because we're using discrete points to model the latent space. We really want to improve this. So again, let's see this quote right here. It is impossible to ensure that the encoder will organize latent space in a smart way compatible with a generative process. Yes, exactly. Because there's gaps here between the different classes, and this is bad because we are not able to generate things if we plug in things here for the latent space. So because of that, we are now moving on to an even more powerful technique called variational autoencoders. Now, this is where things get fun. There's the key innovation here, is that attributes are now, instead of being discrete values, they're now probability distributions. And this is what makes this so powerful. It makes it so the latent space is continuous, and it lets us use the decoder as generators for new data. So now, instead of having a discrete value for the smile, like we did before in the normal autoencoders, now we're using probability distributions for it to represent this. And now the latent space is continuous. So for example, now we encode this guy, smile, gets encoded as decently smiling. And you can think of the standard deviation of these distributions as how confident the model is that it has that property. So in this case, the smile distribution, it's decently high. It's very confident that it's around this point. If it's wider, the model is less confident that this property is true. Anything that's really close, it's less confident. It's very confident about. Anything with higher spread out, anything that's more spread out, bigger standard deviation, it is not as confident in its choice. So with that, there is still downsides, unfortunately. So basically, despite the benefits that this offers as a generative model, now we're going to have a much better latent space. But the issue is that the attributes are still entangled, and the latent space doesn't make sense. Again, this isn't what's actually in the latent space. It would be really nice if this was what's in the latent space, but that's what we're trying to build towards, is to actually have these all disentangled. It does not disentangle these into separate attributes that we understand as humans by ourselves. It instead tangles them all up. And that is what we're trying to avoid. So let's take a look now at what the land space of a VAE looks like. Now we can see things starting to get interesting. Everything groups up around this center, right? So let's go take a look. This is what it looks like without being, this is what it looks like as a normal autoencoder. This is what the land space looks like. But now if we move over to land space of a VAE instead, we can see that it's much more continuous. And the other good thing is that, so everything groups up around the center now, which is good. We actually have access to this, but we can also use this as a generative model. If we sample any point in this box right here, we're going to get something that at least looks like a number, right? So this is what makes this generative, right? So in this case, points like right here, where before we wouldn't have had any idea what this was, now because the land space is continuous, we're able to sample this and get what looks like a half 9, half 1 object, right? So we have the ability now to actually generate things that are basically combinations of classes in this land space. And again, notice that the dimensions are still entangled. Z1 and Z2 don't necessarily correspond to any innate properties of these numbers that we can understand as humans. As we scroll along this axis right here, the Z1 axis and the Z2 axis, it's not like we notice like, oh, this has this special property along this axis that I can control. No, we don't have access to that. We have no idea what this means. It's all entangled still. And that's what makes this unfortunate, is we want to be able to have it so there's individual properties that we can identify with, like thickness or something like that, or position. And we're going to see that in the next example, that we're going to move towards a model that's even better than VAEs. But first, let's talk about the implementation of VAEs. And I know we're going to try and stay away from the math just a little bit, because the math is probably not, I question my own ability to explain the math in a way that would make sense to people, especially for people who don't have mathematical backgrounds in machine learning. So I'm going to kind of try and gloss over the math just a little bit. But essentially, the way that you implement a VAE is, instead of a normal autoencoder, now we have the mean and the variance as essentially, we're parameterizing a normal distribution. So for those of you who, does everyone know what a normal distribution is from statistics? It's literally just the curve that looks like, it's the curves that, what's that?
The bell curve.
Yeah, the bell curve. It's the bell curve, exactly. It's also called a Gaussian distribution, a normal distribution. It's just curves that look like this. And there's when we say parameterize, we mean what two numbers what can we describe this curve with? Well, the curve can be described with two numbers. The first number is, where is this curve on the x-axis? What is the position of that curve? That is called the mean. And then the second thing is, how wide is this curve? And that's the standard deviation that we're parameterizing with. So we can describe these curves in two separate numbers, and we can see those in the formula for the curve, how the curve is described. And those two numbers are sigma here for the standard deviation and mu for the mean, right? So we are able to describe that curve with two numbers. Now, we want the output of our encoder to be one of these curves, right? Well, how do we do that in a machine learning model? Simple. We basically create two distinct layers at the end of our encoder, one that represents the dimensions for the standard deviations, and one that represents the dimensions for the means. Now, keep in mind that this is not just a single dimensional This is not, so in this case, we're showing off each of the dimensions independently as its own curve. But in reality, this can be like a 10-dimensional normal distribution, right? If we combine all of these into one, which is normally what we like to think of, but we can think of them like this as all in their own curves in separate dimensions too, right? So mu1 and sigma1 describe the bell curve for the first dimension in the latent space, and mu2 and sigma2 describe the curve for the second dimension, right? So essentially what we do to actually generate is we sample from these distributions. We do random sampling. I'm not actually going to cover how the backpropagation works for that, because there's something called the reparameterization trick, which we're not going to get into. And here I talk just a bit about the reconstruction and how the mathematical model for the loss function works. Essentially, we need a loss function to tell it how well it's done after every single pass. And the way that we do the loss function, for a normal autoencoder, the loss function is literally just, we ask it, how well does this output face match this input face? And the goal is to have them match up as perfectly as possible. And the better these faces match, that means the better it's done. Yes. Did someone say something?
Very cool.
Thank you, Taha. I appreciate it. So we want the faces to match up as perfectly as possible. So that is called the reconstruction loss, because ideally we're reconstructing the input from the output from the input, right? So the worse we reconstruct, the higher our reconstruction losses. We want to minimize the reconstruction loss. That's how normal autoencoders work. But we're going to see that variational autoencoders include a new term in this loss function to tell us how well we've done. So the reconstruction loss, that's pretty straightforward. We understand that, right? That's the input data and the output data. And we want these to be as close as possible to minimize this reconstruction loss term. But now we have this other thing, this KLD loss term. Now, essentially the KLD loss term, it's the, I'm not even going to be able to pronounce it, but it's the, I'm not going to be able to pronounce what the KLD loss is. It's like Kullbaur-Leibbecker divergence. And essentially what all this term does is measure the difference between two different probability distributions, okay? So when we think about probability distributions, we're talking about these guys, right? Just our normal bell curves. Now, where the reconstruction loss measures how the difference between the input and output, the KLD loss measures the difference between two of these curves. So you see these curves, we basically are measuring how different are these curves. We're not measuring the difference between these curves. We're measuring the difference between our output curve and what we're calling our posterior distribution. And in our case, our posterior distribution is just a normal distribution. And the goal is to map our distribution and force it to look like a normal distribution. And because that's essentially how we're forcing this to be continuous, right? We're basically telling it, hey, you better have this, you better force your mu's and your sigmas to match up to a normal distribution. And that way we can make the latent space continuous. It doesn't just magically become, we don't just get these layers to magically become layers that can represent a distribution, right? We actually have to force them using this KLD term, right? And that's going to make it so now the latent space is continuous. We can sample from the latent space. This is a much better generative model. But still, keep in mind, this is a flawed generative model. We can still generate really weird stuff. There's no intuition behind what this Z1 dimension is and what this Z2 dimension is. Only the machine learning model understands why it put this here. We as humans, this doesn't make any sense to us. As we go along the two axes, it's not like there's this property that corresponds to each axis, which is our end goal. We want axes to correspond to properties that we can control. So moving forward from there, let's talk about beta VAE, which is, again, now this is the third model that we're going to be looking at, and this makes our model even more powerful. So all beta VAE does is add this one scalar term in front of our KLD loss. And what this does is essentially it will constrain the dimensions in the land space and make it so it's a much harder to represent things in the land space. And essentially, this makes it so this actually kind of forces disentanglement of dimensions, which is really cool that we can do this with a single scalar term on this KLD loss. Now, if we actually, I could read this out, but essentially, when beta is 1, it's just the same as normal VAE, right? Because that's just, it's 1, we're multiplying it by the KLD loss, right? But when beta is greater than 1, it applies even stronger constraints on the representation capacity of our land dimension. And then for conditionally independent generative factors, keeping them disentangled. Yeah, so basically, long story short, it makes more sense. The best representation of something is always going to be something that humans can understand. The best way to represent this face is always going to be with a smile, skin tone, gender, beard, glasses, hair color, right? So it's always going to be something that humans can understand. And when we make beta bigger and we make this KLD term more important, it's basically it's forcing it to try as hard as it possibly can to get those human understandable dimensions, right? And the way that this works is, so therefore higher beta encourages more efficient latent encoding and further encourages disentanglement. However, of course, this is going to have a negative impact on the reconstruction quality, because as we force the importance of the KLD loss, we're going to basically be giving our decoder less room to work with, and make it harder for it to understand how to properly reconstruct the model. And thus, there is a trade-off: higher beta, more disentangled dimensions, but unfortunately, not as good reconstruction. So there's a trade-off there, right? As there are with most hyperparameters. And this is the first hyperparameter of our model that we have control over, is this beta term. So moving on, this is really cool. This is where we actually get to start seeing beta VAE disentanglement results and how this works. So long story short, we have these dots, right? There's these dots here, and this is our training data, right? And beta VAE has separated out the position, the Y position, the X position, the scale, the rotation of our dot as separate properties in the latent dimension. So our dot has these properties, and beta VAE has learned these properties and put these in various dimensions, right? So we can see that the position corresponds to the second latent dimension very well, the Y position, right? And as the Y position increases, as we increase the value in the second latent dimension, it actually corresponds to the position of this dot, which is really cool. As we increase the value in the sixth dimension, we can see that it is increasing the X position of it as well. So it's not completely disentangled. We can see that rotation still somewhat correlates to this. We can see that this guy has a different rotation than this guy down here. And we can also see that the scale kind of makes it a square instead for some reason. But what's cool is the fact that this actually corresponds pretty well to these human understandable dimensions, to these human understandable properties of this little box, right? Now, let's talk about what the actual issue is with this. And the biggest issue with this is the fact that we don't get to pick these properties at all. None of this, we don't have any say over what it discovers as human readable properties, and we don't have any, and there's still small amounts of correlation between what should be independent attributes. We can see that the rotation is entangled with the scale still in the first dimension, and we can see that rotation is somewhat correlated with position as well. Ideally, we don't want that. We want no correlation between separate attributes, and we also want first of all, we want to pick what properties we have, right? We want to be able to say, hey, my data has this property that I want to actually train for, and I also want to pick what dimension I put it in so I can manipulate that specific dimension. Here we can see that you actually have to analyze the dimensions, and it throws it in random dimensions for you, right? For some reason, when they trained this one, it picked, hey, we're going to put the Y position in model 2, and it's going to put the X position in dimension 6, not model 2, dimension 2. I'm going to put a scale in dimension 1, right? So we can see that basically arbitrarily assigns human understandable properties to arbitrary dimensions that it discovers, and then because of that, we don't actually have any say over what dimension is selected, and we don't have to say over what properties we're actually training for. So this is really cool because now we're actually separating out properties of the data into individual dimensions that we have control over, but we actually have to do analysis to figure out what dimension has what property, and we don't have any say over it. So because of that, we are now going to move on to the final and most powerful version, which is what we are using, which is AR-VAE. Now, AR-VAE basically takes what beta VAE gave us and just makes it omega powerful. Like, we're way more powerful now because we're fixing literally everything that was wrong in the last example with beta VAE. Now, with AR-VAE, we add a new term into the loss function, this regularization term. And we're not actually going to go into what the regularization term does, but essentially, we are now allowed, basically, to choose what dimension we want and what properties we want, as long as we have the data to pass into the model with it. So now we pass in basically the information that we want to be stored in a specific dimension and what dimension we are going to represent the property in. And because of that, it's able to actually learn as it goes what dimension it should store things in and what properties should be represented, right? So that fixes all of the issues of beta VAE right there. And it's all thanks to this final term in the loss function, right? So moving forward, we can actually see how much more powerful AR-VAE is compared to beta VAE. So let's just take a look at some of the entanglement results. Here we can see that in terms of orientation, it's much more powerful because we have, okay, so in this case, let's take a look, at orientation. So that's rotation, right? So in this case, we are rotating this little square guy as slowly, and yes, he rotates. In this one, we can see that when we try in beta VAE, when we try and change the orientation, it does a bunch of weird stuff with the shape instead. It's not a square, it's an oval, it's a heart, it's a square, it's a heart again, and it kind of works on the final ones, but keep in mind that originally this is supposed to be a square, right? For the X position, we can see that it does a much better job. In this one, it's also, the shape is somewhat entangled as well, so we get blobs randomly, right? This guy stays a square the whole way through because it's disentangled. The X position is disentangled from the shape of it. Y position, basically exactly the same case. We can see that as we move through the Y dimension, the Y position dimension, it literally just moves it down the Y position. And in this case, it's changing the shape, which we do not want. Scale is much the same. We can scale it up. In this case, it's actually, we are able to tell it when we pass in the data that we want, what direction we want to enforce the monotonicity in. In this case, beta VAE, we don't get to pick that. So beta VAE actually starts big, goes small. We tell it that we want to start small, go big, and it will listen to that, and we can actually do that. And here as well, you can see that as it gets smaller, the shape gets really distorted. Shape seems to be correlated with like literally everything because you can see how weird it is, right? Here we actually have control over the shape here in a separate dimension, and beta VAE actually, as we can see, kind of just randomly changes the shape around, but there's no real correlation between the monotonicity of the shape, right? Anyway, moving forward. So this is a comparison of the different architectures that we've just gone over. So autoencoders, the land space is discontinuous. There's like no disentanglement. It's unsupervised, and there's no improvement. But VAE, it's continuous. Now the land space is continuous. It's a good generative model. Its disentanglement is still low, which makes it really hard to actually mess with the properties you want to generate new things. It's unsupervised, but the continuous land space at least allows for generation between known classes. Beta VAE, continuous, good disentanglement, unsupervised. It enforces the constraint on land space, and this improves disentanglement. But then, finally, AR-VAE, it makes it so the land space is continuous and monotonic, and then basically now we're able to actually supervise what dimensions we want the properties to be in and what their values are throughout it, right? So this basically lets us, choose what dimension we want to store each attribute in, as well as what attributes are actually end up end up being stored. So finally, now we're back to that's the presentation on. Does anyone have any questions about these VAE models before we move on? All right, no VAE questions. Nice. Moving forward to hyperparameters. So the hyperparameters are the parameters that we're passing in to the model that are going to be impacting our training metrics. The distinction in machine learning between normal parameters and hyperparameters is that parameters are what the model is training and learning. So that's like the weights in the model, the biases in the model. The hyperparameters are parameters that you pass in at the start of the training process, and they stay the same the entire time. So let's go through. So we're interested in three types of hyperparameters: model hyperparameters, training hyperparameters, and loss hyperparameters. The model hyperparameters basically specify the shape of the machine learning model. This is the number of layers, the type of layers that we're using, and the size of the latent space. And now keep in mind how the latent space works. That is, again, just the really compressed the number of compressed dimensions in between the encoder and decoder. So basically, we have access to how many of these layers there are, how wide these layers are, what type of layers these are, and that's the model hyperparameters. It can basically controls everything about the model. Then onto the training hyperparameters. Training hyperparameters basically control the training process. So that's the batch size. That's the amount of data that we take in at each time step. Let's do with a bit of the back propagation, which we're not going to get into today. And the number of epochs, which is how many times we pass the entire training data set through to have the model learn from it. And then finally, the loss hyperparameters, which are the three variables within the loss function here. So we have the beta, which controls the KLD loss, and gamma, which controls the amount that we care about separating into individual dimensions, or how much we care about controlling those individual dimensions. Beta controls how much the separation actually occurs, right? So moving forward here, essentially, yeah, the model type. In our case, we're using LSTMs, but we could also plug in. It's plug and play, right? You can literally just take an RNN, throw that in there as well, test it out, see if it works. Model layers, of course, the number of and size of the layers in the decoder and encoder. Again, this potentially affects all metrics. LSTMs are actually better than RNNs for what we've seen, but there's a lot to play around with there. Latent dimensions, that's the number of dimensions, latent space. Essentially, if we squeeze them harder, it can lead to lower reconstruction accuracy. It's better just to boost the beta, is what we found. Batch size. So the batch size is the number of data samples that we run through the network before the computation and backpropagation. So low batch size, basically you speed up training at the cost of fine-grained training accuracy. This also impacts all of the batch size can theoretically impact all of your metrics. The number of epochs, that's basically just how many times you pass the data set through. Basically, if all the other hyperparameters are good, it means that more epochs is always going to be better. It's going to yield better accuracy. But you risk, if they're bad, you can overfit. You can potentially screw things up. And accuracy can actually go down. You can overfit significantly if you do too many epochs, right? So finally, the loss hyperparameters. The beta, again, that's the importance of the KLD loss term. High beta means good disentanglement, but this is at the cost of reconstruction accuracy. Gamma is the importance of the attribute regularization. High gamma basically enforces the monotonicity disentanglement of the desired dimensions. And this is, again, at the cost of accuracy. You're probably noticing a pattern here. We can do stuff, but the trade-off is almost always accuracy. And then finally, delta is the spread of the posterior. We found that as long as delta isn't too low or crazy, it's not that big of a deal. It doesn't really affect our case. So into the model training and metrics. So model them basically. This is the big guy, right? This is what's actually going on. Basically, so we pass in the data set and we pass in the hyperparameters. And what ends up happening is we split this up into three different steps, right? There's a pre-processing phase where we have to pre-process the data that's coming into a form that our model can read. Then we train the model. Essentially what happens is the data goes through the model here, and we can see the model here, right? We have the encoder, the mean, and the variance. We sample it into the latent space, and then we decode it, right? And then we get the reconstruction loss, we get the KLD loss, and we get the latent samples, and we use that to calculate the final loss, which back propagates through the model, and that is the model training sequence, right? So then we also, and keep in mind that the training hyperparameters, that all affects the loss computation up here. And in the end, once the model has been trained, we take the fully trained model and then we run metric computations on it to figure out how well the model is actually done, right? So let's move forward and just discuss this briefly. Essentially what happens is before we actually pass the DNA sequences in, we need to convert it to a form that our model can understand, right? Keep in mind, this is a computer. We can't just pass in a string and expect the model to understand it. We do something called one-hot encoding. And what one-hot encoding does is it takes our classes and basically creates a matrix that represents the position of our letters, right? So for example, the DNA string AATG would be converted into this matrix here, where the first letter in the matrix, the first row in the matrix, this is a column, not a row. The first column in the matrix represents A's. If there's an A in that position, we mark it with a 1. Everything else is a 0. Then again, we do another one. 1 in this position, 0s in the rest. T's, we mark 1 in the T position, 0s in the rest. G, we mark 1 in the G position, 0s in the rest. So this is a very common machine learning technique for class-based machine learning. You see this a lot. You have to do one-hot encoding for your data. And in our case, this is what we're feeding in. So that's pre-processing. Now let's go over the actual model training, and we're not going to get too deep into this because this can get really complicated, and this is where I spend most of my time head deep in the model code, suffering and trying to fix things. So essentially what's going on here is we have the reconstruction loss, right? And we can actually see, if you recall the reconstruction loss from before, we can actually see the reconstruction loss going through and being computed here, right? We have all of the individual terms. Here's the reconstruction loss, here's the KLD loss, here's the regularization loss. All of our hyperparameters are coming in. We have beta coming in to moderate the KLD loss. We have gamma and delta coming in to moderate the attribute regularization loss. And even if we follow the arrows, we can see everything going in, right? We have the encode training sequences, and we have the output sequences, and we're comparing them in the reconstruction loss and getting our final value. And then we sum these all up to get our final total loss, which gets fed back into the model for our backpropagation input. And with that, we, in the end, arrive at a fully trained VAE model. So let's talk about the metrics that we're using briefly. So we have, first of all, validation accuracy, which is how accurate the model is at reconstructing the sequences, right? That should sound familiar. That's kind of what the reconstruction loss manages, right? We want the outputs to match the inputs as much as possible for it to be a good decoder. So the way that we do that is we basically just compare the inputs and outputs and see how close they are and how well they reconstruct. We're actually I just had a meeting about this today, and we're thinking about redoing this metric, but it's important for an autoencoder to be able to accurately reconstruct its input. So that's the first thing we care about. We care about our validation accuracy. Next up is our disentanglement metrics. The disentanglement metrics talk about how many of the attributes are being represented in each dimension of latent space. Again, we want very low disentanglement, because if we have very low disentanglement, we are able to basically manipulate whatever attributes we want and construct a completely new thing from that latent space, right? So that's why the disentanglement is so important. We want very good disentanglement. And then finally, the property correlation metrics. And this is, we basically figure out how well a property is being represented by a dimension in the latent space, which is, we also care about that, right? We want it, so we want the property that we want to be highly represented in the latent space. We want there to be a good positive correlation there. So that's the metrics that we actually really care about, right? And we're computing all of those in afterwards. So moving forward here, finally, let's talk about the grid search. And essentially what the grid search does is we take in all of the models and we take in all of the metrics. So after every single model gets trained, we throw it into the model and metric database. And then we pass those into a visualizer, which lets us, we're basically then able to look at the metrics as humans and figure out what model is the best. And then from that, we can select the optimal model to use as our generative model that we're actually going to be using to design those DNA sequences. So visualizing the metrics, we literally visualize grid searches as grid search, as a grid. There's an application for this. It may be linked on, I don't know if I have it up in, let's see, cd visual backslash visualizer. Let's see if I can get the Python 3 module up right now. What? I was just running this. What do you mean?
How can you do this to us, James?
What do they mean? I don't understand. I'm in the right folder. Right? Python 3 MHTTP server. Address is already in use, which means I have somehow accidentally... It's up already. Okay, so we literally represent our grid search as a grid. This is why at least I think it's called a grid search, is because you literally have a grid of parameters. We have these are hyperparameters, the gamma, the beta, you might remember these. And there's also another parameter here that we've added recently that's not in this presentation called the dropout. And essentially, we can see that we have a bunch of different metrics, if we click through this, that we can basically look at this and determine how well our different models have done with different hyperparameter combinations through the grid search, right? So this is how we visualize our results with a grid search. So going back to our presentation, basically, yes, the grid search can be viewed in the form of an actual grid, and this lets us, as the humans in the loop, determine from the metrics how well different parameters impact different metrics that we care about. And ideally, the best combination is always going to be high accuracy, high correlation, and high disentanglement. And we want the best of all three of those. Of course, it's not that easy. There's no set number of parameters that actually give you all three of those. It's a trade-off. The higher you get your correlation and disentanglement, the lower your accuracy is going to be, sometimes to the great detriment of ourselves. But basically, us as the data mining and management lab, you can see Professor Bogdanov, Alex, and me sitting here, and we look at this and we basically try and determine from these results what the best possible model is going to be. Again, sometimes we might say, okay, let's take a look at the most powerful model in terms of the correlation. Let's take a look at the model with the best accuracy. Let's take a look at the model with the best disentanglement. And then we'll pick what model we think is going to be the best and which trade-offs are worthwhile. And with that, we then actually take that model, and then we are able to use that model to synthesize and generate. So let's take a look at the actual process of synthesis and generation. We take that optimal model, and then we take our potentially arbitrarily selected sequences and our desired attributes, and we put all of that together. And in the end, we are able to actually generate sequences with those properties. So the way that this works is we take our optimal model, we extract let's first talk about the encoder extraction path. So we take the encoder, we take arbitrary or potentially selected sequences, and then we encode those sequences, and that gives us a nice representation of the sequence with the properties that we want. Now we manipulate the properties in the lane space that we want, aka wavelength and chemical stability and brightness. And because we're using AR-VAE, we have the ability to freely manipulate those. Hopefully, if the model has properly disentangled them and represented them in the right dimensions, which, because we're using AR-VAE, we can try and force that. And then we basically manipulate that, and then we pass those sequences into the decoder, and the decoder is what reconstructs a sequence from those desired properties. And then we have basically the decoder will output those sequences, and then we finally, in the end, synthesize them, and that's basically it. Then we have, ideally, with this model, we have reconstructed DNA sequences with our desired properties that, when synthesized, will create genomic silver clusters that are in the near-IR spectrum. So, I actually have stuff that talks about this, right? So, essentially, since the model is AR-VAE, we have the guarantees about the lane space. It's continuous, the attributes are encoded along our desired dimensions, and the dimensions that we've encoded these in are monotonic. So again, with this in mind, we can manually push up or manipulate the values in the dimensions to get to our desired attributes, right? So that is basically everything. That's the presentation. Does anyone have any questions about what has gone on here, particularly the AR-VAE model, because that's the most exciting thing? Yeah, that was a lot to take in. Absolutely. There's it's very heavy stuff. I know. There's
Massive brain size.
Yeah, it took me like a solid, I want to say like 3 or 4 weeks to understand what was going on when I joined this project.
I'm still digesting the title.
I'm sure most of us are, and that's how I was digesting the title for a few days when I joined as well. It's not an easy, it's definitely not an easy research process to understand, but we can break it down. We are using a machine learning model to generate special nanoparticles that fluoresce a certain color, and this is the process by which we do that, the machine learning model, right? So it's definitely a mouthful, and this is if you say the title to someone without any context, it can severely scare them, as it has many people. Which is probably why the attendance is down tonight. I applaud all of you who came to this talk knowing that this was the title, and knowing that despite the fact that you didn't probably understand any of this at the beginning, it still came anyway to try and learn what it did mean. So, any questions about any of this? If not, we can wrap things up. I'm glad it was only 9:15. June, you can end the recording so I can give the